I'm going to talk about brain stimulation in the brain. And at the beginning, I will show the concept of what I mean with brain stimulation. It's quite a broad field. And how this relates to the virtual brain. And then the second part of my talk, I will give some examples. Will be rather superficial, so if you have questions or so, then please interrupt if you want to know details. First of all, I think the general question is how does the brain work? So this is what we want to know. That's why we do neuroscience, and this is one standard paradigm where we consider the subject, the patient, as a black box or gray box, and we do measurements while we control the environment, and then by correlating the measurement with the stimulation, so that's the control over the environment, we try to gain insights into the subject, so to say what's going on in the brain. So we want to find out the relationship between input-output, what is the response to stimulation, like a stimuli. That can be behavior, but the input can be also stimulation technique. But the problem is now, of course, if you control the environment in a way that there's no stimuli, the brain, you will always measure something, so the brain is always active. So which means that's actually conditional, so this input-output relationship you would need to relate to the given state of the brain, okay? So in this, if you understand this better, I think this will have an impact on diagnosis and therapy. Okay. So what do I mean with brain, or with stimulation, brain stimulation? So on one side, there are brain stimulation techniques. They can be intracranial, so direct stimulation, electrical stimulation. It can be also transcranial stimulation. So one is invasive, the other one is non-invasive. And so the invasive one, for example, if you do brain surgery, that's a kind of a standard way of inferring the functional property of the neural tissue that you're penetrating, stimulating. And there's evidence that there's a link between the transcranial stimulation and the direct electrical stimulation. That's here you see on the right side a work by Fox et al. That compare the efficacy of stimulation techniques, the intracranial as well as then transcranial, and they relate this basically to resting state networks, and here it's listed simply for different disorders, so the therapeutic effects. So just to make a link between the different stimulation types, and there is a hope that maybe in the future we can, so to say, get rid of the invasive stimulation and use transcranial stimulation schemes. So in there, I think the virtual brain can help. So now I will show a little bit about that. Another stimulation type, questions. So if you, okay. Another type of stimulation, sensory stimulation, and can be visual, can be auditory, and so on and so forth. So there's a way of interacting with brain rhythms. So if you stimulate with a temporal pattern, like very rhythmically, and then we can actually change the intrinsic rhythm of the brain, like the alpha rhythm that's shown here. So if you stimulate around this intrinsic frequency, which is on the x-axis, then the stimulation frequency is on the y-axis, then you see that there's a range when you stimulate around this intrinsic rhythm, that you can change the intrinsic rhythm, you can move it. And this kind of plateau that then is shown here in the graph is a kind of a sign of flexibility of the brain to follow the stimuli. And this has been also clinically been used as a sign for healthiness. And for example, epileptic patients, you can provoke epileptic discharges, for example. So how we bring these different types of stimulation into the virtual brain, that's basically what Misha showed. So we use MRI imaging MRI data, diffusion, and structural data. We use the pipeline to get it into TVB, and then the result is then a description of the geometry, of the folding of the cortical surface, divided into brain regions. This, of course, depends on the postulation. And then two types of connections can be defined. So one is homogeneous, so this is the nearest neighbor connectivity between these neural masses on the cortical surface. I don't know if you see it, so this is tessellated. So you have this mesh of triangles, and then this short-range connectivity is homogeneous. It only depends on the distance, and it's basically describing the connectivity of these neural masses that are at each network node with the vicinity. And the other type is coming from the diffusion MRI data that is called large-scale connectivity that is heterogeneous. So it's really specific, connecting brain areas, different brain areas in the brain. Okay, so that's a repetition, I think. Questions? Fine. So the basic concept is that the brain is a network, of interacting neural masses. So these are the smallest elements. As a basis, we use the structure of the brain, so structure measurements, and as we assume local dynamics. I will come later back to that. And the thesis, so the main theme behind is that the function is constrained, shaped by the structure. So we assume that the structure is constant within a certain time period, let's say an hour. In terms of stimulation, so I would say stimulation creates causality. If there's something circular, so then stimulation is something we have under control, and we can look at how the model, so this virtual brain is responding. And the other point is that the brain, the TVB, the model, is agnostic for the type of stimulation. So it doesn't matter if it's sensory or a stimulation technique like TAX or so. The model has to deal with all of these different stimulation types. So, and then the result basically should be a validation, so reproduction of experimental data, providing mechanisms, that's very important, and then maybe more important are the predictions. So this is then something that we can use to design future experiments. So now I'm going to show you here how to stimulate the virtual brain. And how the TVB responds to focal stimulation and to TDCS, so transcranial direct current stimulation, so with a constant. So what TVB does, so I showed you this slide before, but now I opened the box. Right, so now you see here mathematical descriptions inside. So TVB is opening this box, and now we have description of the dynamics of the state of the brain on one side, and then something that I call observer system. It's in the TVB in the GUI called monitors. So a forward model that relates the brain state, so the dynamics, to the measurement. So that's then really specific for the measure you're looking at, so EEG, MEG, fMRI, and so on and so forth. Yeah. And so far we have, I talked about the structure of the brain. Here I start now talking about dynamics. So we have to decide about the local dynamics. This is maybe something worth to mention, that in TVB we assume that the dynamics are not in the links directly. There are time delays in the long-range connectivity, but mainly there are no differential equation, no real propagation in the physical sense. We have the dynamics in the virtual brain. We assume that the dynamics are in the nodes, so in the brain areas. So, and for this, as shown before lunch, there are different models available. You can classify them differently. So here, just listed a few of them. So some of them are very simple and generic, generic 2D oscillators. Some are more biologically informed. Okay, and then your response, so to say, of a stimulation depends also on the type of model you use. This is something that is up to the user to decide. Okay, all these different models are implemented in TVB, and there's a mathematical hierarchy behind that looks like that. I don't want to scare you away with all this math. Just to walk you shortly through, as you have to decide in the first level about variables that you're interested in. So firing rate, current, postsynaptic potentials. And on the second level, you connect them, so how they're related. And then on the level of here, this neural mass network level. So if you consider different populations, excitatory population, inhibitory, interneurons, pyramidal cells, something like that, then you can create a local network. So here is only t is time. So there's no space in, it's only temporal. Then on the next level, so to say, you can come up with a field description where you add space and then the connectivity. So here the homogeneous short-range connections, then, and the other term is heterogeneous, this is the long-range connection. So this is then the heterogeneous, the long-range connections are then also the reason why we have to simulate it. We cannot compute this analytically. So, okay, that's then in TVB, this awful equation is, so to say, discretized and then for the simulation. And what I want to point out here, that actually very natural in the description of TVB, we have the stimulation. So everything is actually defined based on so-called impulse response functions. So this, what I highlight here in red are the stimulations. So in physics we say perturbation. So that's the input, so to say. The behavior on each level is described by the input. Questions? So how to stimulate in TVB? We had this already shortly in the hands-on. So first we have the network state. Okay, I define the network state as the activity in the large-scale network, and we can stimulate in two different ways. We can stimulate different brain regions, but we can also perform a surface stimulation. And then there are different requirements I'm going to show you. We can also change over the period of the simulation. So you already simulated the TVB. If you perform a change in the parameters, and then you make a branching, so then this is actually similar to perturbation or stimulation as well. Then you would mimic then, let's say, a stroke or something like that. Okay. So coming to region stimulation, so the requirements, of course, are the long-range connectivity. So that's the connector. And then, first of all, you have seen this page before, so that's the TVB GUI. What you have to specify or to decide on are the temporal, the shape of the stimulus. So it can be something linear, can be Gaussian. So linear would be related to a constant parameter change or like a transcranial direct current stimulation, sinusoidal stimulation like for deep brain stimulation, or pulse train RTMS, for example. So surface, oops. Ah, yeah, okay. And okay, that's time. And then in space, that's this button here. And in the GUI, then you define, so to say, what which areas to stimulate, and then you can select the brain areas and give a scaling how the weight, so to say, of the stimulation for different brain areas. Surface stimulation require the cortical surface reconstructed, like you see here. Then again, you define, you have the temporal behavior, so linear, sinusoidal, and so on and so forth. And now in space, it's like you can decide for, I don't know if you can see that here, like needles. You can place needles on the cortical surface and then define in space the behavior of the stimulation. So for example, a Gaussian, so then you have the spread here of 4 in the frontal pole, left and right hemisphere. So here's one, there's another one, here and here. And then you can simulate how the stimulation would look like. And then you end up over time defining the, having here a Gaussian temporal course, then the spread, which is also Gaussian in space then of the stimulation. Question? It's fine, okay. To give you an example, that's what Renzi showed before. That surface stimulation where you use a very simple oscillator. This is something that is, let's say, mimicking then in the response a event-related potential. So it's a damped oscillation. Okay, so it's a spiral so something two-dimensional. And then what's nice here, then you see the distribution of the stimulation in space. Okay, and you can, so to say, you stimulate, and you can look in different brain regions how, if there's a response or not, and how this is spreading. Okay, questions so far? All good? Not sleeping? Okay, now I move on to examples. So I will show you the focal stimulation in human and then in mice, and at the end the TDCS stimulation in human. So that's the human brain model. And so again, this picture, we have a reconstruction of the cortical surface, division into brain areas, then the distinction between short-range connections and long-range connections. Here in this model study, I simply stimulate all the nodes within the brain area. So I stimulate one brain area, and then under the assumption of very simple dynamics, which is like this 2D oscillator, this damped oscillation, I'm looking at the brain responses, so to say here, okay? And then analyzing the brain responses with respect to the different stimulations. So this stimulation type is closest to direct electrical stimulation in the brain. So if you would use the GUI, then the stimulation, the temporal equation would be something like an alpha function. So it's a very short peak, yeah, and in time. And then in space are these focal points. So within all, setting this up within focal points within all the brain areas. Within one brain area, all the nodes within one brain area. Yep.
I'm just not familiar. What does alpha come from?
Alpha, it's like an exponential function multiplied with it's a mathematical, okay, you have a linear function and an exponential function, and the sum of both, or the multiplication, is basically the alpha function. It's a linear rise, and then you have an exponential decay. That's the definition of an alpha function. Sorry. This has been used to describe visual stimuli. So for Jensen and Ritt in the 90s, they used this for the cortex. Because they were not having the subcortex, the thalamus, they were using the alpha function to mimic the visual input. You're welcome. So to summarize the model, so this virtual human brain, we have, so this you've seen before, short-range connections are homogeneous, long-range connection heterogeneous. We are dealing here with more than 16,000 neural masses. So these are these points on the grid. So each point is kind of covering 16 square millimeters, of which we have these neural masses are divided into brain areas, 190, of which then 116 are subcortical. So this is the standard model actually in TVB, what you can use right away. And so now we stimulate the one brain region and looking at the response. So what we basically see, we have the stimulation here. And this is the response over time in 3 different brain regions. Premotor cortex, dorsolateral, then cingulate posterior, parietal cortex medial. And what you see that response is, so to say, different in strength and in how long they last. So the decay time is different. And if you look at topography, so here are the 3 different stimulation sites, the premotor cortex, cingulate, and parietal cortex. You see here over time the lateral view of the left hemisphere. That's here, here, and here. And then the medial bank of the left hemisphere. These are the stimulation sites in red. And then from this on, we see how, so to say, the brain is responding. And what's interesting here is that after a certain time, these are distant, so that's lateral, that's medial, that the activation pattern looks similar compared to the neighbors. So these are neighbors in the medial bank, but the response is completely different. So this is interesting because here in the medial bank we cannot, we would need to use deep brain electrodes to evoke this pattern if we are interested in this pattern. But we can also go or let's say cortical, transcranial to stimulate this brain area then to obtain the same activation pattern. This is the standard TVB average brain. So this is, let's say, a proof of concept that I'm showing here. It's not individual data. It's an average brain. So so this is one example for 3 stimulation sites. I've done this, we've done this systematically. We for all the brain areas, we vary the ratio of short-range to long-range connections because it's interesting to know to which extent information is processed on the short range or on the long range. And then also, we vary the range of short-range connections So this is the nearest neighbor connection between 1 centimeter and 4 centimeters. So this ended up with here 36,000 per meter configurations. I had a cluster for that, so it took setting up the whole thing, putting it on the cluster, and then I came back after 1 week and I had the results. So by then, actually, you have to analyze it as well, and so in the we are interested in these response networks, and basically to extract the characteristic responses, we perform the decomposition method. So it's not only simulating it, it's then also you end up with a lot of data, you have to analyze it. Okay, and the result is actually summarized in these two tables, where you see on the y-axis here, these are the functional resting state networks. And then these are the stimulation sites in the columns that, so if it's red, then the stimulation, if you stimulate here, let's say S1, then you trigger something, a pattern that looks like the memory network at rest. So I did a comparison of the responsive networks with the resting state patterns, and then I looked for the significant matches, and these are summarized in these tables for the given set of these 8 resting state networks. That's for the cortex, that's for the subcortex. What's interesting for the cortex is, let's take the memory network, that the stimulation sites are actually scattered all over the brain, so that, which means there are different ways of addressing, triggering the same activation pattern. Okay, to summarize this study, spatial proximity is not a good predictor for the outcome. We see different function networks, and function network can be induced by stimulating different sites that can be scattered all over the brain. And we find back the experimentally known resting state networks. So how to test this? This is a simulation study. It's like performing an experiment on a highly complicated model. So I think only patient data would be good here. So ECOG would be a possibility. Then going stereotactic EEG, deep brain stimulation. So this would be the way. So then we would end up with epileptic patients, for example, or then Parkinson patients. Another way is, okay.
One question regarding the definition of a resting state network. You would expect it to be, for example, the motor network would be one network, right?
Yeah.
So, but stimulating the hand knob with TMS does something completely different compared to stimulating the foot area or the face area.
Yeah. You see this postulation here, it's quite rough. I mean, you don't really have the foot or the extremities kind of resolved in the postulation. So you would need there some good information to divide this better. So this is simply too rough, and this is...
it would be possible to implement the homunculus is well known.
I would say so. Yeah.
Conceptual question. So, what TVB needs to set up the model is just a structural connectome and some simulation parameters, correct?
Yeah.
And now at that level, when you simulate data, you compare now to real data. That's what.
Yeah. My understanding from before was that also you can use functional data to tune your model. Yes.
What's the general strategy?
There are different ways. I mean, what I've done here is, because this is all based on, I mean, it's similar to a Brodmann postulation. And if you go to the resting state literature, they describe the patterns also in terms of Brodmann. So I could use this as a kind of an interface to have masks, so to say, for the different resting state networks in this postulation scheme. And then on that basis, I was comparing it. So it's not the case that I was having really real data, resting state data. Another way would be functional connectivity, or to compare that. Is this answering your question? Okay. Oops. Okay, so that's how to test it. So what we've done is to go on to the mouse brain. So we used, we exploited the database from the Allen Institute. So this is a database of tracer studies. So in 3D, they code the injection sites, and then how they are then connected to all the others brain areas in the brain. So what you see here, basically, that's one slice of the database, of the Allen database, and you have the postulation of in colors. These are different brain regions. And then you may not see it. These are all little dots, actually. And these little dots that are here, in, let's say, within the big blue area here, or cream blue, they all belong to one structure, and they all represent one. So each dot is one tracer study. So, and based on that, on this database, I was first reconstructing the isocortex. And then because we have the connectivity, so we have the postulation, which is then projected here on top of the surface, and we know how these brain areas are connected. Yeah, the diffusion after injecting the tracer, waiting a certain time, then where they end up. This tells us about the connectivity, and this is then basically here then this heterogeneous connectivity which is directed. So giving us a directed information how area 1 is connected to 3 in other areas. And on top, then again, seeing the short range connections, which is homogeneous. And these two types of connectivity are coded in these matrices. So for short-range connections, we have the weights between source nodes and target nodes. And for the long-range connections as well. In addition, we have then the time delays. Because the tracer studies, you're not reconstructing the fiber tracks. Okay, we use here as a proxy the Euclidean distance. So it's maybe for mice less critical compared to human, because it's simply the surface is smooth. Okay, and then we performed the same stimulation scheme. So we stimulated the brain areas and looking at the responses. Okay. Sorry for that. To summarize the model, it's comparable to human. So here we talk about 14,000 neural masses. So for human, we had 16,000 roughly. The postulation is finer. So here we consider more than 500 areas, of which are actually 400 are cortical. So you see already here that the pronunciation is actually more on the subcortex than on the cortex. So for human, it's usually the other way around. The responsive patterns, so I cluster them, and we end up with 12 motifs. So motive 1 to 6 and 7 to 12, and they are spanned by 3 patterns. So pattern 1, 2, and 3. These motives are independent, and but still they are correlated. So here in blue, I highlighted the correlated patterns, and they actually reflect functional networks. So this would be something like a saliency network and the default mode networks. So these are the two that I could that we could find back, less the somatosensory patterns. And this, of course, we can relate to functional data, so in vivo, from mice, so with using voltage-sensitive data, so of the cortex. So you have here the cortex. So you open the skull, you look from the top, and then you see here the right hemisphere. And this data from Canada, from Majid Mojerani's lab in Lethbridge, and they stimulated the whisker, auditory, visual, and so on and so forth. And then they looked using this technique, the spatial temporal response in the brain. And this is something that we are using right now to match, so to say, model and experimental data. Questions for that?
Back to the human model.
Mm-hmm.
Did you test the effect of different stimulation parameters on the brain responses?
With stimulation parameters, you mean the where?
You do the alpha model.
Yeah.
But as you said, there are different
Mm-hmm.
functions.
Yeah, I mean, okay, I'm an electrical engineer by training, and the kind of from control theory, you can, so to say, it says that you can determine the response of something that is stable for a certain time by giving an impulse to the system, and that's basically this alpha function. So you get the brain response, and
You mentioned DBS, you have a sign function.
Yes. Then we talk about synchronization, entrainment phenomena. This is definitely highly interesting, and this is upcoming, I would say. I mean, this is what, of course, we can extend. This is a starting point, and yeah.
The question to the local connectivity in the Allen-Brain
Yeah.
database.
Where
I
is
didn't
it?
quite get it. So this is, you took
Ah,
this
yeah.
from experimental data from the database, or
Yeah.
you also...
This is theoretically, this is assumed. So here, it's actually based on Amut Schütz's work on sheep there with Breitenberg. They published a book about statistics in mice and about the local connectivity. And here I varied they came actually up with this notion that it's exponential on the cortex. So here, out of technical reason, I used a Gaussian kernel. And I'm varying the, so to say, the standard deviation, the local range for the short range connections, the spatial range for the short range connection between, I think, 0.5 millimeter and 1 millimeter, something like that. Yeah.
So does the stimulation always evoke the same responses?
Depends on where you stimulate. I mean, if you
Stimulate the same place, would that
yes,
always?
yeah, because the model is very simple. It's dead when there's, so to say, if you are not stimulating, then you see a zero line.
The intensity of the stimulation vary?
I tested that. Not really. It's more than it matters. It has an impact on its scale, so to say, the response, I would say.
If the neural avalanche is right, then one event can cause different type of cascade.
Mm-hmm.
Some are small, some are mass. So this is different from actually what we know about the real brain.
So if you talk about avalanches and cascades, I would, this model is too simplistic. So you would need a multi-stable models. Yeah, multi-stability. Then to then really have a cascade of let's say from a switch from down to up states and so on. Okay.
What about the ongoing activity in the mouse brain? Do you simulate that as well?
Okay.
Synchrony between different parts.
Okay, here yeah, that's a good question. Okay, I consider this is the impulse response, yeah, and if you consider an impulse as simply a realization of a noise process, then it's like an ongoing. I mean, you should see it also an ongoing noisy data. So I haven't done it for mice, but for human. So you simply drive the same model with noise, and then you see the same patterns emerging, but then alternating over time. Okay.
How many inhibitory neurons and excitatory neurons do you have used in each area? Or the question was also in the other direction to local connectivity or to the architecture of a microcircuit and the interconnection of microcircuits.
Mm-hmm.
It's critical.
Yeah.
Okay.
Here, this model is very simple. You have one parameter that is, let's say, you can call it criticality, you can call it excitability. It's, so to say, determining the ratio of inhibition to excitation. In mathematical terms, it's the damping factor. You can also, I don't know, I think I don't have time. We run out of time, right? Okay, maybe shortly. I will. So for the simulating transcranial direct current stimulation, we use a way more sophisticated local model. Okay, first of all, what is not in TVB, we have a forward model from the. Are you familiar with transcranial direct current stimulation? Okay, so we have these huge sponge electrodes on the scalp, and what we calculated here based on a finite element model actually that we then used in the virtual brain. We calculate the current density within the head, yeah, and then we calculated, so to say, the orthogonals of the currents that are orthogonal to the cortical surface and use this as input. This may be similar to what you have in neural navigation with TMS. So then based on these maps, you adjust the intensity of the TMS. But now here, the local model is way more sophisticated because we want to describe EG data. So here we distinguish between interneurons and protecting neurons, so pyramidal cells. Interacting with excitatory and inhibitory interneurons. So that's the Jensen-Ritt model. And the stimulation here is, we decided to only let the pyramidal cells be affected by the TDCS. And then the output, so to say, is then what we project using the observer system, so the forward model, into EEG. Okay. And so here we have a way more, a microcircuit, so to say, or something that is more kind of canonical, let's say, of a cortical element, of a cortical column. And so in TVB, we would use in temporal equation simply a linear constant. So that's just a linear equation would be then, so to say, the offset. And the region scaling, this is something you don't have in TVB. This comes then from this finite element forward solution. Okay? So this is not directly set. This is something you cannot do in the GUI. You need to use the command line plus the information from the finite element model. And doing that, simulated the scalp EEG, so what you see here. So these are the spectra, frequency spectra of the scalp EEG, and then the functional connectivity. And what we basically see that there is increased functional connectivity under the anode, and also an acceleration of the rhythms under the anode, while overall there is a deceleration. So this is something that the model predicts, and you would like to test then experimentally. So to summarize, can use the basic are large-scale brain networks of the human brain. We assume temporal dynamics, so this is up to the user. Then, so we are able to reproduce functional data, functional connectivity, and then the predictions are basically based on or regarding the rhythms, the dominant rhythm, the increase under the or around the anode, and the rhythms are getting more harmonic, and an overall deceleration. So the take-home message is that I think this TVB, large-scale brain modeling, is helpful for understanding what's going on in the brain, that there are explorations that you can only do in silico, like the stimulation of all the brain areas. You don't want to do this in patients or animals. And still they are doing this in silico. These are models, toys. We can reproduce to a certain extent functional data and come up with mechanisms and predictions that then we can also translate, so to say, from between mice and human. And I think for the future, I showed only average brains that we can. That this type of modeling supports personalized medicine using very individual models. Okay, thank you very much.